The last video here on paper chromatography is going to talk about the experiment itself. You're going to have a piece of these paper that you can get from the instructor bench. The first thing is you're going to split that paper into the different ions that you're going to run on the paper chromatography experiment. So in this case, there's 5 ions. You might have all 5 ions or you might not have one of them. It just depends on what the stockroom provides. And then there's a line that's called a known line or the mixture line. That one is just a line that contains all of the ions mixed together. And then there's gonna be 2 samples of unknowns, but they're really the same unknown. The only difference between these 2 unknowns is that one is spotted twice, the other one is spotted 4 times. And I'll talk more about that in a little bit. One thing I want to point out here is when you're writing these things down, you make sure they're all done in pencil so that they don't actually interfere with the experiment itself. The line from the bottom to the top here could be between 1 to 1.5 centimeters. Your sample is going to come in this form right here. What the stockroom has done is provided all the different ions name on it, and then there's also something called a mix, which remember is all of the ions combined together. Through here you can see that there's some of these small tubes. Those are called capillary tubes. Here's what it looks like. The capillary tube is these guys right here. They're inside the actual test tube. When you take them out, hold them, you see that there's some solution inside the capillary tube already. That's because of capillary action by water, so it climbs up a little bit into the glass. You actually only need maybe half of that to spot onto your paper. Take that capillary tube and just hold it upright and then put a spot on each of the ions, and then the mixture sample, and then your unknown sample. The size of this spot shouldn't be too big or too small. Once it gets too big, it might run into the other lane and then it might contaminate the other sample. Here's how you're going to apply the sample. I recommend holding the capillary tubing upright and just dip it, hold it, push it down so the sample goes to the paper. You don't need to do it too hard. The water would transfer to the paper pretty quickly, and you don't want to have too much of it goes in there. The known sample has to be spotted 3 times. Make sure you spot it and then dry it first. You can dry it by waving the paper around, air dry it, or a heat lamp in the different parts of the lab. You can just put the paper underneath a heat lamp. That's also going to dry up the sample, and then come back and spot it again a second time, and then dry it again, come back and spot it a third time. That's how you're going to spot multiple spots. You don't just spot them all 3 times right away. Same thing with the ones where you have to spot 2 times and 4 times for the unknown. Now let me repeat that again about the unknown. The unknown is actually the same unknown. Let's say you pick unknown number 1. You're going to spot for this sample, you're going to spot it twice. For this sample, you're going to spot it 4 times. So that's the only difference between these 2. It's not that you're going to pick one unknown here and another unknown here. It's the same unknown. They're just spotted 2 different ways, because in case something happens with this one, you have this one as a backup, or if something happens with this one, you have that one as a backup. So the solution you're going to use to actually run it, the solvent system, is this solvent system right here. So it's not water, but it's a mixture of some organic solvent. You're going to pour that solvent into a beaker so that it just barely covers the paper at the bottom, and then you put the paper in there. Paper has to be rolled up looking like this, and you notice here that the sample is on the outside. Put a tape on the top and maybe tape in the middle so that it would stay like a cylinder together in that solution. But you notice that the solvent is very low here, right? And then you just cover it up with parafilm, and that thing should slowly rise up. That's going to be what the experiment is. When it's done, as soon as you take that paper out, draw that solvent line so you know where that solvent is. Take that piece of paper and just drop it into a developing solution, which is going to be in a tray in one of the fume hoods. As soon as you developed it, you should see a bunch of different dots. Example shown in this particular paper chromatogram. So you see all these different dots corresponding to different ions. This is done with a different solvent system, so it's not the one that you're going to see, but it would look something similar to this. So once you have that, you just need to dry your paper, take a picture of it, and then measure the distances. Again, the distance would be from the beginning, the starting line is marked here, to the center of each spot, and then you're going to have to calculate your RF, which is dividing that distance by the distance from the beginning to the distance of the solvent. Here's a quick example of one of the spots. For example, let's say I measure the beginning to the center of that spot is 3.05 centimeters. There will be rulers provided for you in the lab. And then I measure from the beginning to the solvent line is 5.33 centimeters. I can divide this number by that number, and that should give me my RF value.